My name is Ruchika, and my research is based on cardiac repair and heart failure. So currently I'm working with induced pluripotent stem cells. So we put them through a 14-day to 22-day protocol to differentiate induced pluripotent stem cells into cardiomyocytes. And we monitor their growth and we monitor their changes just to make sure that we are getting the kind of cell types that we want. And once I obtain the cell types at the end of my protocol, I use those for downstream reactions. So usually I do qPCR to maybe extract RNA and look at the fold change reactions between the different genes of interest. Once we start culturing the cells, we use EVOS on a daily basis to sort of check our cultures, to monitor their progression from one cell type to the other, to see how the differentiation actually occurs. And usually in terms of scientific research, when you're trying to write up reports, it's good to have EVOS because then you have a day-to-day captured image of your cell's progression. Another part is because of the fluorescence imaging. So I usually try out live cell imaging, at least on 5 different day points. And I use those images to sort of monitor my progress. And then it makes it easier to report when you're talking to colleagues or your superiors about your research. I think the NEO model is more user-friendly. It helps you actually navigate better on the screen, so makes for better visualization. And the different channels for fluorescence. So previously there was a lever that you had to use to change from your bright field imaging to your fluorescent imaging, but now it can be done automatically on the screen. So that's a good improvement. Previously we did not have the cell counting feature, and that itself is a massive improvement, especially when you're working with monolayers and you've got multicultures. So if you've got different cell types in the same well, it's easier with the cell counting feature where you can actually differentiate based on circularity or density of staining and everything to differentiate between your different cell lines within the same culture. The NEO models also have the incubator with it, so I can monitor cells over a 24-hour period and create a time lapse to make sure that, yes, these are the minute changes that are happening over a long period of time. I've seen beating cardiomyocytes. So that was the best thing, because when you're culturing cells, you're just going by morphology, you're just going by what you know by studying and research papers that, yeah, this is what they're supposed to look like. But a true form of validation comes when at the end of your 14-day protocol or your 22-day protocol, the actual cardiac cells beat with rhythm, and then that confirms that yes, the cells that you have are cardiomyocytes. And it's easier to visualize on this because you don't disturb the wells too much, because any disturbances would impact their beating or their movement. So with the imaging system, without disturbing them, you can actually visualize their rhythmic movement. I have a goal towards which I'm working. So because my interest is in cardiac repair or heart failure, I know that any research that I do, it might have negative results, but at the same time, it is contributing to the larger field of maybe curing any kind of heart failure or stroke victims. So that is the biggest motivation behind it. But partially also because when you do research, it's easier to have mini goals and achieve those mini goals. So first, you see if you can actually achieve the kind of cells you want. Then you see if those cells behave the way you want. And then you see if those cells actually have the expression of things that you want, which can be applied to clinical science or even medical practice.